Photoreceptor mechanism. Visual or phototransduction is a process by which light energy is converted into receptor potential in visual receptors. Potential changes that initiate action potential in the retina are generated by the action of light on the photosensitive compounds in rods and cones. The conformational change which happens when light falls on these substances trigger a sequence of events that initiate generation of action potential. Ionic basis of the generator potential. In the dark, sodium channels in the outer segments of the photoreceptors are kept open by cyclic GMP. So there is a steady current flow from inner to the outer segments of the rods. The steady current also flows in the synaptic ending of the photoreceptors. So when light falls, it leads to the increased conversion of cyclic GMP into 5-GMP, resulting in the closing of sodium channels. And so that occurs because of the decrease in the concentration of cyclic GMP. So this decreases current flow and resulting hyperpolarization which generates local graded potential, that is generator potential in the synaptic terminal of the photoreceptors. Note that in dark retinine 1 in rhodopsin is present in Leven's configuration. You can see when rhodopsin, when the light falls on it, it gets converted into bathmorhodopsin, lumirhodopsin, metorhodopsin 1, 2, then gets converted into transretinal and scotopsin. And when in case of dark, the scotopsin is going to bind to the 11-cis retinal, which is activated from transretinal, converted into transretinal to form rhodopsin. So here you can see in the disc, when light falls on cis retinine from rhodopsin, it gets converted into transretinine and it gets activated. It is going to activate and help in the production of generator potential in the rod or cone. Development of receptor potential or phototransduction. It is a local graded potential generated in the ganglia cells only. The sequence of events involved in the phototransduction of rods and cones are when light stimulus occurs, there will be structural changes in the retinine 1 of rhodopsin. That is, 11-cis retinine gets converted into all transretinine, which leads to conformational change in the opsin, which causes activation of transducin, that is G protein, that is GT1 in rods and GT2 in case of cones. Alpha sub, because of which alpha subunit separates from the G protein, and there will be activation of the phosphodiesterase, which converts cyclic GMP into 5-GMP. So which causes decreased intracellular concentration of cyclic GMP, which leads to closure of sodium channels, which leads to hyperpolarization of the receptor, that is decreased release of synaptic neurotransmitter glutamate, and so which causes the disinhibition of bipolar cell through interneurons, and thus an action potential develops in the ganglion cells. So this is the mechanism which occurs in case of dark, where the high levels of cyclic AMP levels, they keep sodium channels open, sodium enters in, and which causes depolarization, helps in the release of glutamate neurotransmitter. In case when the light falls, so there will be production of the transducin, which causes the decreased levels of cyclic GMP, so which causes the closure of sodium ion. So sodium levels decreases, which is going to lead to hyperpolarization, and that leads to decrease in the neurotransmitter glutamate, and so these are going to inhibit the interneuron, and interneurons in turn excites the bipolar cells.